Hello, everyone, and welcome to the 20th INFOGEST Viva webinar on food digestion. This is part of our Viva webinar series featuring presentations by PhD candidates and recently graduated PhDs in the field of food science and digestion. These webinars are typically held on the first Wednesday of each month and are organized by a team of postdocs from Teagasc. So today, my colleague Chen and I will be hosting the webinar on behalf of the team. Our presenter today is Cathal Dold, a PhD student based here in Teagasc, Moorepark, and the School of Food and Nutritional Sciences of the University College Cork. Cahall's research employs both in vitro and in vivo approaches to examine how infant milk formula processing will influence protein digestion and also gut barrier health. As part of his research, Cathal investigated the physiological benefits associated with lowering heat treatment during milk formula processing. And his presentation is titled Influence of Infant Milk Formula Processing on Protein Digestion and Gut Barrier Health in Vitro and in Vivo. So without further delay, I'll hand over to our speaker. Cathal, the floor is yours.
Hi everyone, my name is Cathal Dold and I'm a PhD student based in the Teagasc Food Research Centre Moorepark, Ireland and at the School of Food and Nutritional Sciences at University College Cork. Today I'm going to talk about my PhD thesis which looked at the influence of infant milk formula processing on protein digestion and gut barrier health in vitro and in vivo. So the main aim and objective of my thesis was to look at how does infant milk formula processing influence protein digestion and intestinal barrier health. So in chapter 2, I looked at does IMF processing influence growth performance and protein digestibility in the gastrointestinal tract of young pigs. In chapter 3, I looked at does IMF processing affect gut barrier physiology and mucus production in the upper small intestine of these young pigs. As well as this, in chapter 3, I looked at does IMF processing influence digestive and bush border membrane enzyme activity in the upper small intestine of pigs. And finally, I looked at does jejunal gut lumen collected from pigs fed IMF diets alter health biomarkers in an in vitro gut barrier model. So in chapter 1, for my literature review, I looked at the effect of processing infant milk formula on protein digestion and gut barrier health in vitro and preclinical. So the main aim of this literature review was to look at how does IMF processing influence protein digestion and gut barrier health in the infant. So this literature review compared heat-treated IMF versus unheated or reduced heat-treated IMF. And what I found is that heat treatment of IMF results in a reduction in protein hydrolysis, an increase in protein aggregation in the stomach, a reduction in villus height and an increase in crypt depth in the small intestine, and a reduction in goblet cell number in the small intestine, increased intestinal permeability, and heat treatment of IMF reduces brush border membrane enzyme activity in the small intestine. This literature review was published in the Journal of Dairy Science in 2024. So after doing this literature review, I produced an infant milk formula powder by high temperature IMF, seen here in the left on the red, which is the standard method of producing IMF, which involves preheating the milk at 80 degrees for 30 seconds, followed by a final heat treatment at 125 degrees for 5 seconds prior to spray drying. So having looked at all the literature as part of my literature review, I wanted to see could we produce an IMF powder with lower heat treatment. So here on the right in blue is our membrane filtered IMF, which is an alternative method of producing infant milk formula. This involves preheating the milk at 50 degrees prior to using 1.4 and 0.2 micrometer membranes to produce an IMF prior to spray drying. So while the two different processing methods differed, they both had an identical protein, lactose and fat content, Both had a weight to casein ratio of 60:40, which represents a stage 1 infamal formula suitable for infants less than 6 months, and 250 kg of IMF powder was produced. This work was published in 2021 in Dairy. So after producing the two IMFs, I wanted to see, does IMF processing influence protein digestion and gut maturity? So just a brief background. So how did processing impact on IMF properties? So you can see here is that membrane filtered IMF has significantly higher native whey protein content of 59.9% compared to only 4.5% in the high temperature IMF. So while the native whey protein content differ between the two different IMFs, the advanced glycation end product, carboxyl methyl lysine, was similar in both the membrane filtered IMF and the high temperature IMF. So after producing the two different IMFs, I performed a dietary intervention trial using a pig model. So the pigs were aged 28 days at weaning, and the pigs were divided by treatment. So 10 pigs received this high temperature IMF, and another 10 pigs received this membrane filtered IMF. The pigs were fed IMF at a 35% inclusion diet, and the pigs were fed twice daily, once in the morning and once in the evening for 28 days. Aged 56 days, 180 minutes after their final feeding, the pigs were slaughtered, and then gut lumen and tissue samples were collected from the stomach, the duodenum, the jejunum, and the ileum. So then in chapter 2, I looked at how does IMF processing influence growth performance and protein digestion in these young pigs. So the growth performance and feed intake was monitored over 28 days by looking at the pig's average daily feed intake, average daily gain, and their feed conversion efficiency. As well as this, the gastrointestinal contents were analysed for the degree of protein hydrolysis following the OPA method. Protein and peptide size was analysed by size exclusion chromatography, and the free amino acid content was analysed using an amino acid analyzer. And this work was published in 2023 in Food Research International. So did IMF processing influence growth performance in pigs? So these two graphs here show the average daily feed intake in grams per day and the average daily gain of the pigs in grams per day. And what you can see is that at day 7, the membrane filtered IMF fed pigs had significantly higher average daily feed intake and average daily gain. But by over the course of the dietary intervention trial, there was no overall difference in average daily feed intake and average daily gain. So following on from this then, did IMF processing influence protein digestion in these pigs? So this graph here shows the degree of protein hydrolysis measured by the OPA method, and this represents the micromoles of free amino groups released per gram of protein. And what you can see is that in the stomach and the duodenum of pigs fed these membrane filtered IMF diets, there was significantly higher degree of protein hydrolysis compared to the pigs fed these high temperature IMF diets. As well as this, the membrane filtered IMF fed pigs had significantly higher levels of water soluble peptides and proteins in the jejunum and the ileum at sizes 10 to 18 kilodaltons, 18 to 28 kilodaltons, and 28 to 65 kilodaltons compared to the pigs that were fed the high temperature IMF diets. So then following on from this, after seeing how membrane filtered IMF diets significantly increase protein digestibility in the young pigs, I wanted to see how does IMF processing influence intestinal barrier health in the pigs. So the gut lumen, tissue and mucosal scrapings were collected from the duodenum and the jejunum. The gut lumen was analysed for trypsin activity, and the tissue and mucosal scrapings were collected and analysed for villicide, crypt depth, and goblet cells. The brush border membrane enzyme activity was also analysed in these samples, and I also looked at tight junction and mucin expression within the duodenal and jejunal tissue and mucosal scrapings. And this work was published in Food Research International in 2024. So, does processing influence digestive enzyme activity in the upper small intestine of pigs? So after seeing an increase in protein digestibility, I then went on and looked at trypsin activity in the duodenum and jejunum gut lumen. And what I saw is that trypsin activity was significantly increased in the duodenum of the pigs fed these membrane-filtered IMF diets compared to pigs fed high-temperature IMF diets, while there was no difference in trypsin activity in the jejunum. So following on from this then, what difference did I see in gut barrier physiology? So this data here represents the histological analysis I did, and what I found is that processing had no effect on villi height or crypt depth in the pigs. However, when I went looking at the number of goblet cells in the pig's small intestine, what I found is that in the jejunum of pigs fed membrane-filtered IMF diets, there was a significantly higher number of goblet cells compared to pigs fed high-temperature IMF diets. And these goblet cells seen here are involved in mucus secretion. There was no difference between goblet cell number in the duodenum. So then after seeing an increase in these goblet cells which are involved in mucus secretion, I wanted to see did IMF processing type increase mucin protein levels in pigs. So what I did here is I looked at mucin 2 protein expression in the pig's tissue, and what you can see here is that mucin 2 protein was significantly higher in the jejunum of pigs fed these membrane filtered IMF diets compared to the pigs fed high temperature IMF diets. And again, following on from this, looking at this mucus secretion in the pigs, I looked at acidic mucus in the pig's small intestine. And what I did here, this graph represents micrograms of allicin blue per milligram of tissue, and this allicin blue detects acidic mucins within a sample. And what I found is that in the jejunum of pigs fed membrane filtered IMF diets, there was significantly higher levels of acidic mucus compared to pigs fed high temperature IMF diets, with no differences seen in the duodenum. Following on from this, then I also looked at mucin 1, which is a membrane bound mucin, and mucin 2, which is gel forming mucin expression within the duodenum. And what I found is that the pigs fed these membrane filtered IMF diets had significantly higher mucin 1 and mucin 2 gene expression in the duodenum compared to the pigs fed the high temperature IMF diets. So following on from after seeing an increase in trypsin activity and an increase in goblet cell numbers and mucus secretion, I went a step further and looked at how did processing influence brush border membrane enzyme activity. So the brush border membrane is the microvilli covered surface area of the small intestine, and brush border membrane enzymes are involved in the final step of intestinal digestion. So what I did is I extracted the brush border membrane following this protocol by Simona 2023, and I looked at the brush border membrane enzyme activity of different peptidases and lactase. And what I found is that in this graph here, you can see the milliunits of lactase per milligram of protein. And what I found is that the pigs fed the membrane filtered IMF diet had significantly higher brush border lactase activity in the duodenum compared to the pigs on high temperature IMF diets. So then chapter 4 looked at intestinal mucus in vitro response to membrane filtration processing of infant milk formula. So what I did in this chapter is I wanted to see how does jejunal lumen samples from pigs fed IMF influence an in vitro gut barrier model. So what I did here is I digested both the high temperature IMF and the membrane filtered IMF powders following an infant in vitro gastrointestinal digestion model using the INFOGEST method. And I also collected the jejunal gut lumen samples from the pigs fed these two different IMFs. So then what I did is I created an intestinal in vitro barrier model using enterocytes, CACO2 cells, and goblet cells, which is HT29 MTX cells, to mimic the small intestine. So then I differentiated these cells for 21 days to form a polarized monolayer with a trans-epithelial electrical resistance value greater than 800 ohms per centimeter squared. And then on day 21, I exposed the monolayer to both the I60 IMF samples and the jejunal gut lumen samples for 4 hours. And after the 4-hour exposure, I collected a cell lysate. So then I asked, how does jejunal gut lumen from pigs influence the in vitro gut barrier? So what you can see here is a Western blot of Claudin 1 and Ochlanin from the monolayers exposed to the jejunal gut lumen. And you can see from the jejunal gut lumen collected from the pigs fed these membrane filtered IMF diets, Claudin 1 and Ochlanin protein expression were significantly higher compared to the jejunal gut lumen collected from the pigs fed the high temperature IMF diets. So as well as this then, I also saw that the jejunal gut lumen collected from the pigs fed the membrane filtered IMF diets significantly increased mucin 2 and mucin 5AC gene expression in the cell monolayers, but I noticed that there was no difference in protein levels in the monolayers treated with these samples. But then when I took the I60 in vitro digested membrane filtered IMF samples, the mucin 2 protein was significantly higher compared to the high temperature in vitro digested high temperature IMF samples. So after previously from in vivo results and in vitro results, having seen an increase in mucin and goblet cell numbers, I wanted to see, does this membrane filtered IMF protect against pathogenic bacteria? So what I looked at here was Salmonella enterica adhesion. So what I did was I created HT29 MTX monolayers and differentiated them for 21 days. And then on day 21, I treated the monolayers for 4 hours with both the in vitro digested IMF samples and the jejunal gut lumen samples. Then the samples were removed and the monolayers were exposed to Salmonella enterica for 45 minutes. So then I collected the inoculum and the T45 cell lysate samples. And I spread these samples on BHI agar plates to determine the total bacterial counts to get the percentage adhesion. And what I found is that both the in vitro digested membrane filtered IMF and the jejunal gut lumen membrane filtered IMF significantly reduced Salmonella enterica adhesion to the monolayers compared to their respective high temperature IMF samples. So then to conclude, what are the benefits of a membrane filtered infant milk formula? So a membrane filtered infant milk formula more closely resembles human breast milk as it has lower heat treatment and has an increased native whey protein content. Membrane filtered infant milk formula has significantly higher protein digestibility. This membrane filter and inflammatory formula also supports gut barrier maturity with an increase in tight junction integrity, an increase in goblet cell number, an increase in mucus production, and a reduction in pathogenic bacteria adhesion. And finally, a membrane-filtered inflammatory formula offers an alternative choice for both manufacturers and consumers. So I'd just like to take this opportunity to thank my two supervisors, Linda Giblin from Teagasc and Aylin Sahin from UCC, and I'd just like to thank all my different collaborators, and a special thank you to all of you for listening. Thanks very much.
Thank you, Cathal, for your brilliant presentation. Now it's time for question and answer, and I see that we have a question in the Q&A box. There's a question for you, Cathal. Are there also effects of heat treatment on bioactive peptides found?
Yeah, so that's actually a good question. It wasn't covered in these slides, but I've covered it before on other slides. But basically, so for the pig samples that we have, we didn't look at any peptides in them. That'd be something I'd hope to do in the future if I had more time. But previously in vitro samples collected from Aqua2 monolayers treated with IMF powders, in the apical samples, we synthesized some MEM IMF peptides and we treated them on our HT29 MTX monolayers, those individual peptides. And what we found with one or two of those peptides is there was mucin gene expression increased, and with one peptide in particular then as well, we found that mucin protein was increased after 4-hour exposure to one of those MEM IMF peptides. So we didn't actually look at the heat-treated IMF peptides, but we just focused on the membrane-filtered IMF peptides and looked at mucus bioactivities.
Right. Thank you. We have another question for you, and it's about how is it possible to translate these results obtained from the weaned pigs to neonates, the infants, before solid food transition?
So yeah, it's a good, like that again. That'd be a next stepping stone if you were to go further with those with a dietary intervention in young babies from birth. That basically, but you have to go one step at a time from in vitro to pigs, and that was all I had time for in the 4 years of my PhD. But ultimately, then as well, if I was to go on and do that, then as well, you'd like to look at breast milk samples in comparison with the two different IMF powders. But again, before you even go to that, you'd want to start with that breast milk as a control with your in vitro studies, and then your pig dietary intervention before you go on to those babies.
Interesting. That was an interesting question indeed. I was interested with your results on the adhesion of salmonella to the cell line. What could be the reason why IMF has less bacterial adhesion to the small intestines compared to heat-treated milk?
Like that was one of the salmonella adhesion experiment. That was one of the final experiments I did for my last chapter. So basically, what led me to do that was we were seeing that mucus increase as you went along with both in vitro results and then the in vivo results with the pigs. So with that mucus layer providing like a protective layer on the cell or the gut barrier, we wanted to see then if that protective layer was preventing adhesion of pathogenic salmonella in this case. But I suppose again, if I only had time to do one pathogen, so I went with salmonella, but you could also look at E. coli or Chronobacter, or flip that again, you could look at do either of those IMF powders increase the adhesion of probiotic strains like lactobacillus or bifidobacterium strains.
So basically you're saying the higher the mucus layer you have possibly the better protection against.
Adhesion of those pathogens,
Yeah.
yeah.
Okay, interesting. Okay. You did mention that you looked at carboxymethyl lysine in the infant milk formula powders. Did you look at any other advanced glycation end products?
No, in my two IMF powders, the carboxymethyl lysine is the only glycation end product that we measured, and that was actually done in Denmark at the University of Copenhagen. But I suppose again, if you were producing those IMFs again, you'd probably look at other glycation end products like furazine as an example that was measured by another group in ENRA, and they measured at different stages of production, and they produced similar IMF powders to us. So again, if we were to do it again, we'd measure furazine and carboxymethyl lysine at that different stage of processing, because like both the powders were spray dried, and that's obviously going to contribute to the glycation formation. So what we'd like to do is, before processing, even in the milk, and then during processing in the liquid IMF, measure the glycation end products, and then again after spray drying in the final IMF powder. And even like people go on then and look at the storage effect, like Maillard reaction is going to happen during storage, so even measure it or monthly in a year or two after or whatever.
And these end glycation products would adversely affect the height of the villi and all those components you measured, or how?
No, with that, like I did some preliminary studies where I took synthetic glycation end products. I got synthetic carboxymethyl lysine and furazine and carboxylethyl lysine, and we put them down our gut barrier, and we saw like an effect on tight junction expression and mucin expression. And there's also another group, I think it's Wu et al. Is a paper there that they looked at synthetic peptides down, similar to us, looked at synthetic peptides as well down in the in vitro gut barrier, and saw an effect on tear and tight junction expression as well.
Just checking if we have one in the Q&A box. We have none, but I do have one more. In terms of microbial safety, how does this membrane filtered infant milk compare to ordinary heat-treated milk?
Yeah, so I suppose that's one of the most, before you get on to anywhere, that's probably the most important question to look at. So like both IMF powders we produced were safe, and like they met the microbial safety requirements outlined as it's European Emission Regulation 2073, I think, from 2005. And what they outline is the requirements for the microbiological criteria of foodstuff. So in our IMF powders, going by those guidelines, we had no pathogenic bacteria detected, so there was no Corynebacter, Sakazaki, or Salmonella. But the only thing is, our membrane filtered IMF powders, they did have higher bacterial loads than the high temperature IMF powder. So in the MEM IMF powder, I think there was 10 to the 7 colony forming units per gram of powder, and in the high temperature then there was only 10 to the 2 CFUs per gram of powder. So like the membrane filtered IMF powder, that was produced at pilot plant scale, so it was using a discontinuous processing system, which like that left the IMF vulnerable then to contamination after filtration. So in theory, in an ideal world, if you were upscaling that to an industrial setting, that MEM IMF powder would be produced using a continuous processing line under like strictly controlled, regulated conditions.
And so this microbial load was determined before spray drying or after spray drying? Because
no,
the spray dryer has a heat component as well.
Yeah, but those colony or those CFUs were in the final powder.
Yeah.
So maybe that's the thing as well that you could look at is before going into the spray dryer or different stages of the processing. Take samples for that as well.
Yeah. Okay. I don't know, Daniela, would you have a question for Cathal?
Oh, I think he answered all questions very well. Thanks very much, Cathal. That was an excellent presentation.
Thank you, guys.
Right, so we come to the end of our webinar. Cathal, thank you very much for your lovely talk and for this live and question and answer segment. I'm aware that in a week or so, you're defending your PhD. Best wishes with that.
Thank you.
And to our viewers, we hope to see you in March, where we'll be having Connor Fitzpatrick, again from Teagasc, presenting his PhD work. For now, it's goodbye.